Welcome to Ask the Expert. Ask the Expert is a brief, informative, engaging, and a lively discussion with cutting-edge science and advancements in type 1 diabetes research. And we're hoping it's a new style of scientific communication, sort of like more of a town hall than a lecture. It's very short. It's time sensitive because we know everyone is super busy in their laboratory and all their other commitments. And we just really believe that, hearing from all these different scientific voices who have, interest in studying type 1 diabetes will lead to an increase in the knowledge base. So today we're really pleased and lucky to present Dr. Lorenzo Pasquali. He's coming to us to the University of Pomepeu Fabra, Madrid in Spain. And so it's sort of late for him, but so thanks again for joining us. And his title, the talk what he's going to talk about is beta cell non-coding regulatory functions in type 1 diabetes. So just a little background on him. He's a professor in genetics and the group leader at the University of Pompeu in Fabra, Barcelona, where he leads a multidisciplinary team of experienced computational biologists, as well as researchers with a primary expertise in experimental genomics and chromatin biology. He received his medical degree and specialized in pediatrics at Gaslini Hospital in Genoa, Italy. And then he trained in molecular biology at University of Pittsburgh and in transcriptional regulation and computational genomics at IBAPS. I don't know if you guys say that differently, in Barcelona. His scientific interest is understanding the genomic regulation of pancreatic islets and its implications to human diseases. And the outcome of his line of investigation has resulted in milestone publications that has really opened the path to understand the genomic regulations of the islets and shed a light on the molecular mechanisms that underlie type 1 and type 2. And as we've heard from several other people, scientists, basically a lot of the defects and mutations that are being seen in type 1 diabetes are now found in non-coding regions. So this is a very important line of inquiry. His lab also is really involved in developing tools like UMI for cats. That's an R package that allows processing, analysis, and visualization of data obtained by UMI4C. That's a technique that enables to characterize and quantify 3D chromatin contacts. And also his lab put together the Eyelid Regulum browser. That's a web tool that allows really fast access and exploration of pancreatic islet regulatory genomics data produced by all different labs worldwide. So we totally laud that idea of collaboration. And so yeah, welcome. And we're happy to have you.
Well, thank you very much, Monique, for all this, for the introduction and for the invite. I think it's really nice. Like the format is innovative. It's a nice way to share our knowledge in a different way than the normal conferences or meetings.
Yeah. Would you like to?
So as you said, well, our lab is really a hybrid lab. So we are doing molecular biology on one side, and then analyze our experiment and doing bioinformatics. We have a team of bioinformaticians that works and try to understand and disentangle all these large genomic data sets. And myself, I'm as a background, I'm a medical doctor, so I've been dealing with type 1 diabetic patients in the past. Now I'm in a completely different field, of course, but I think this helps me to keep a link with the patient, with the hospital somehow. Yeah, so I prepared just very few slides in a very simple way, just to explain our research line more than explaining most of the experimental work in detail.
Yeah, that's perfect.
Okay, great. So I can, let me see if I.
I think we just need to do the, yeah, there it is. Perfect.
It's perfect. So exactly, this is the title. We are really centered our effort, our research question in understanding the non-coding part the genome in the beta cells. And I think we really have two main research lines in the lab. So on one side, we are trying to understand what happens to a beta cell under environmental stress. And this is something that is related to type 1 diabetes. Here we really try to understand the regulatory mechanisms that are central to metabolic diseases, glucose metabolic diseases in general. Then we have another research line in which we're studying these very rare tumors that develop from the beta cells. So we aim in studying these rare tumors because we think that trying to understand what is changing in the beta cell identity could allow us to understand more about the physiology of the beta cells, and eventually finding also some drug targets that could in which we could benefit for diabetes itself. Perhaps if we understand what goes wrong in this tumor, perhaps this can help us to understand what to do better in other diseases that affect the beta cells. But let me talk more about the first research line. So as a general background, here we have this. Well, of course, the fact that most of the association that is linked to type 1 diabetes uncovered through these genome-wide association studies are showing that many of the variants that are associated to the disease are falling in the non-coding part of the genome. This is something really that is common to most complex diseases. So this is true indeed as well for type 2 diabetes. And it gives an extra challenge because having all these variants in the non-coding part of the genome makes it more difficult to link them to the transcriptional changes. And indeed, in this sense, what is really useful and what it had been done in the field for diabetes as well as other complex diseases was that of trying to map regulatory elements, non-coding RNAs across the genome, and indeed this can help to identify some of the variants that are associated to the disease and are functional and are possibly affecting the regulation in a cell-specific manner, indeed, because these regulatory maps, they tend to be very tissue-specific, but as well also state-specific.
Can I just ask a question? When you talk about the cis-regulome, are you including sort of the methylome? I mean, is this included too?
Right. We didn't work a lot on methylation, but that indeed has a lot of regulatory function, the DNA methylation itself. Mm-hmm. And while regions that are less methylated indeed were shown to be more active and coincide with active regulatory elements. So this includes indeed the mapping of other features such as histone modifications or chromatin accessibility that helps us to identify these regulatory elements and build these regulome maps that are cell type specific indeed. So this had been done, and indeed it was shown that variants associated to type 2 diabetes and fasting glycemia, they tend to overlap regulums or regulatory maps that are active in the beta cells. And while for type 1 diabetes, this was not true. The variants associated with type 1 diabetes, they're not particularly enriched in the beta cell regulome. This makes sense. This is an autoimmune disease, and most of these variants or many of these variants are really overlapping regulatory elements that are specific to immune cell types.
Yeah.
This makes a lot of sense.
Yeah, it does.
So what we're studying, what we're doing in general in the lab is trying to expose the islets, pancreatic islets, to an inflammatory environment. I think ideally would be really interesting to try to reconstruct the first, or at least the initial stages of type 1 diabetes, although it's difficult. For the moment, what we did was exposing pancreatic islet to different cocktails of cytokines, and then try to map the regulome again. So again, here, as I was mentioning, what we're looking at is are the histone modification, chromatin accessibility. Those are the 3D chromatin structure. So these loops that put in proximity distal regulatory elements with the targets, the promoter targets of these regulatory elements. So by doing these experiments, what we're seeing is that the regulome of the pancreatic islet is actually quite dynamic. So an external stimulus is able to activate regulatory elements in the genome, but not only that, it seems like it's also inducing changes in the 3D chromatin structure, so that these newly activated regulatory elements are getting in contact with their target genes.
And is this specific to the beta cell, this response, or do you see this in, many different cell types, the alpha cell types, the delta cell types? I'm
Right.
just in the islet environment.
Right. So that's something something really unexplored, I think. So the plasticity of the alpha cells or other specific cell types in the islet has to be or is not really understood yet in its details in the non-coding part of the gene. So what we've been focusing was on the beta cells themselves, but indeed in alpha cells or delta cells or other cell types composing the pancreatic islet would be very interesting to map and to see. Actually, there are single cell experiments that are being doing. Kyle Golden is working on this. Has some really nice data that on single cell, and this showing as well that these regulatory maps are possibly changing. So this is indeed very exciting.
Yeah, it really is. And it's really hard I think, to get a grasp on this, because it is so, it's 3D dependent. It's not just sort of one and done, like a gene to,
Right.
gene to RNA to protein situation.
Right, and these 3D chromatin structure are highly complex, so that the same regulatory element, the same enhancer, it doesn't have just a one-to-one relationship. But it can actually are experiments demonstrating that can activate that multiple genes, or that indeed you have multiple enhancers, multiple regulatory elements acting on the same gene. So it's really a highly complex system.
I would also say, and correct me if I'm wrong, but it seems like learning something here in this environment could have purchase, on many other disease states and other, sort of working models of the way this works.
Absolutely. I mean, what we're seeing here for type 1 diabetes could be translated to many other diseases, and other glucose metabolism diseases that is not just type 1 diabetes or type 2 diabetes.
So it's a very fertile frontier. A lot of room for inquiry and new scientific, exploration.
I think so, yes, and much to explore here.
Yeah.
Indeed, this also has to do. I mean, it can be linked to the genetics, which is something that can be done in the sense that if, as I just explained, I mean, the variants associated with type 1 diabetes are not overlapping more than there is no enrichment for overlapping more than the regulatory elements of a healthy and untreated pancreatic islet. What we're seeing is that these newly activated regulatory elements that are activated upon an exposure to this external stimuli these are indeed more enriched for variants associated with type 1 diabetes.
Yeah.
So this is just, I mean, it's an initial observation, it's a proof of principle, much is to do to understand really what is meaning, what is the meaning of this in terms of the mechanisms that are behind all this. But it supports the idea, the possibility that some variants associated with type 1 diabetes are acting through the beta cell, but are acting through regulatory elements that are just silent in normal conditions. And,
can you walk us through the path that a cytokine would take to access the regulatory elements in the nucleus?
Well, that's another these are other interesting questions actually that should be disentangled in their details, because well, indeed the cytokines will act on receptors on the beta cell surface, and then these will act through different signaling pathways, and eventually will activate transcription factors that will act in the nucleus of the cell and bind the DNA in order to somehow activate some regulatory elements. This is probably, I mean, in a very naive way, what is likely happening. What we are seeing, well, this is all inferred through sequence analysis and some ChIP-seq data. Is that indeed we have some inflammatory transcription factors. Well, some transcription factors that are typically activated upon a pro-inflammatory stimulus. These are like NF-kappa beta, IRF, but also STATs, and these are we're seeing that are induced and are binding the DNA. Something that we're seeing that is, I think, very interesting is that they seem to bind in combination with very tissue-specific transcription factors. So they tend to bind to this newly activated regulatory element in combination with transcription factors that are typical of that are typically active in a beta cell, PDX1, MAF-A, or NKX6.1. But again, these are initial experiments, and the actual mechanism that is behind all these, and the mechanistic activation of these regulatory elements is still to be elucidated.
Yeah, that's really interesting. Has anybody ever, this is sort of a random question, but has anyone looked at the cytoplasmic vaults as a way that some of the cytokines or messaging could access the nucleus? I asked that because we're talking to a group next week that is investigating these ribosomal vaults.
Okay, I see.
They're very obscure, but you wonder, because they're involved in drug resistance and breast cancer and things like that. And you sort of like in altered environments, they play a role.
Yeah, good question. I don't think this was much explored really.
Yeah, I don't think so yet. But we'll talk with the vault people next week, so we'll hear what they have to say.
That's a good question for for the group. Yeah, definitely. So overall, in very general terms, this is our hypothesis, or what we think is happening is that as you can see here on the top of this model, what we're showing are the variants that are associated through genome-wide association study to type 1 diabetes. These are variants that indeed are overlapping in general regulatory elements that are active in the immune cell types. But what we are observing is that some of them are overlapping regulatory elements that are silent, that are not active in resting beta cells, but can be activated upon the exposure to a pro-inflammatory environment. These enhancers that are the hit of these variants associated type 1 diabetes, they tend to be linked to genes that have function involved in apoptosis, immune response, and beta cell stress. So this would be a different scenario that is what is happening for type 2 diabetes, in which variants associated to the disease are instead overlapping constitutive regulatory elements that are controlling insulin secretion, glucose sensitivity, or beta cell identity. This is our working hypothesis, our model, and around this we're trying to elaborate experimental designs to prove this hypothesis.
What other groups in Europe are kind of focused in on this right now?
Well, we've been collaborating a lot with the ACI indeed, and I think was a very nice and open collaboration with the group. They are world experts in this in the cytokine exposure, knowing all the possible cytokine cocktails that are that can be exposed to the beta cell, the environment that are more stressful to the beta cells. So so with them we had a very important and very strong collaboration.
Yeah, that's great.
But indeed it's a very open field, and indeed has many questions to be answered.
Yeah, agreed.
Not just the genetics, it's also the mechanism, and there's the transcription factor binding and the 3D chromatin structure.
Yeah, no, it's a huge frontier.
So other things that we try to do is to collaborate with other groups and to share our data indeed. So in this sense, this is how we started working on the Eyelid Regulon browser. It was initially just a figure of a paper, and then it became something bigger and bigger, and we started using it to share the data with other groups. Now we're trying to maintain it, and because we know that the Eyelid community is using it and is taking advantage of it.
Yeah, no, this is really excellent. And so if someone wants to just use it, you know, as a first-time user, how easy is it? And I can see that you've got your UCSC up there, WashU.
Yeah,
so people come in and they could either add their data, right? Or they could actually, you know, kind of go through the repository. How does it work for the first time?
So the web tool is completely open. You don't even have to register to use it, to access it. And you can browse through the data. Here you can see chromatin maps and enhancer annotation or transcription factors. Here there are some different data sets that can be loaded. It's not possible at the moment to load your own data, but we're trying to have different data source from different papers and different data sets. If you have indeed some data sets that you would like to visualize, or you would like other people to browse through, please contact me, just drop me an email. We would be more than happy to have the data, your data there, and have it accessed for anyone, and have it integrated with other sources. So I think this is the main idea of the browser, that you can really integrate different data source from different groups and different type of data. In the sense that we have transcription factors, we have regulatory element annotations, and as well as 3D, the annotation of the 3D chromatin structure through this virtual 4C-seq data. So
that's great. I mean, it's human. Well. I think it also has the advantage that it's human islets, right? So that's the real deal. So
no, obviously this it has to be. I mean, we are not trying to compete with any other browser. This is specifically centered on the beta cells, and we try, I mean, to have to have something that is different in its visualization, and that is centered on the pancreatic islets, and that it has, I think, or we hope, it has like a very simple or at least intuitive way to integrate the data, so that maybe like just a genetist that is not like a bioinformatics that wants to approach this because wants just to browse his favorite gene can easily go and see the data. Indeed, if you don't like the visualization and you're more used to look at the UCSC, like the bottom and the top of the eyelid regulum up here, redirects all the data directly to the UCSC browser and to the typical visualization that you have in the UCSC, and indeed the integration with all the annotations from this browser. Or what you need for the 3D drawing.
Okay, yeah. It seems like this invites interdisciplinary scientists too, and to see like, okay, they're working in Parkinson's or whatever, and they found something, and they could kind of come in and cross compare like what's happening in eyelids, or like, you know, closer disease like RA.
Yes, well, yeah. I mean, obviously that can that is open to any science also of any field, of course, to see what if their favorite gene what is doing in the pancreatic islet.
Yeah.
Yeah.
Well, it's fantastic. This is a great initiative. It really is. I mean, it's just so you know.
Thanks.
It's really great that it's a human eyelid, you know, browser, and it's great that it's open to everyone too.
Thanks. So, well, the other tool we are developing are more directed to bioinformaticians. So that's well, as you mentioned at the beginning, we are also developing tools such as the UMI4C or UMI4CATS, which is a tool to analyze 4C-seq data. So again, this is something more specific for bioinformaticians. So in general, obviously, we are used to look at the DNA in a linear way. We know that this is not the case. It's much more complex. But in this complex 3D chromatin structure, there are tools that allows us to identify the loops and the contacts between the gene stretches, and indeed a way to identify these contacts from molecular biology point of view is these 4C-seq experiments, which are then that can then be analyzed, or several tools to analyze the 4C-seq data. But one specific problem that you have with 4C-seq data is that it's not really quantitative, because it goes through different rounds of DNA amplification. So this is a limitation. I mean, if you are comparing two conditions, can be a limitation. So it was developed some from a group in Israel, from Amos group, a technique which is called UMI4C. They also developed some bioinformatic tools to analyze these data. What we did was just to ready to make a package, an R package that helps bioinformation to get the data, to analyze it, to find a way to find peaks or contacts in the genome in a quantitative way. and to have also nice visualization of the locus. So this is what this UMI for cats is about.
Yeah, no, that seems very handy tool for sort of before and after exposure to cytokines.
Yeah, no, well, obviously we use this for before and after cytokines. We needed to have something that was quantitative, so we spent a lot of energy in the lab and to develop these tools. So we thought that this could be helpful for people in other fields and different questions.
Can I ask you, when you guys look at the, the islet cells, the beta cells from different patients, for instance, do you see these same signatures, over and over when you expose the cells to the same cocktail of cytokines?
Right. So, yeah. So we, unfortunately, I mean, this is, these are complex experiments, and also like getting the human pancreatic islet is difficult. So it's not that we can repeat these in, or we couldn't repeat this in a very large cohort of pancreatic islets, but between those that we analyzed, like 4 or 5 samples, for example, we did these experiments that the consistency was very high.
Yeah.
And then of course some of these experiments we did on cell lines, which is it's not ideal for many reasons, but it's very handy from a molecular biology point of view, obviously.
Yeah, you got to start somewhere.
Yeah. But yes, no. Well, we try to use all the signal that were consistent across different samples. So
It would be great if you could get sort of a crowd science situation going on where many labs could look, and use this R package to analyze the chromatin contact profiles, and kind of like all dump that into a repository somewhere and see what it looked like. That'd be really cool.
Yes. Our problem, some of the problems are really, well, from the molecular biology experiment is tricky. So that is something that, well, I mean, that you need some time for the experiment to reproduce it.
Yeah.
So I think that's the limitation.
Well, again, you got to start somewhere. So, and you guys are doing it, so it's fantastic.
So acknowledge the people. This is the most important, because I'm talking for the work of many here, and it's a great group of people really. So here, Mireia, she's a postdoc in the lab, and she's purely bioinformatician, as well as Mark, and Georgina, she just started, and we have other people on the wet lab side, such as Bea, and Clara and Angel. So most of a lot of this work was done by Mireia, and all the wet lab experiments are done by Bea and Clara. So it's a lot of work. So it's really great to have such an enthusiastic team.
Yeah, it was fantastic. I mean, it's great to see so many young scientists, and all kind of, it seems like a great group, and you're really working together to crank out some excellent new ways of thinking about the eyelid biology. I think it really is, you guys are really on the forefront, along with the Galton group. You're really digging into these complex problems. And so we celebrate you guys and salute you.
Thanks a lot.
Yeah, for sure. I'm going to open it up as there's anyone in the audience would like to ask a question. Oh,
Just,
no, jumped off. I asked plenty, so I hope I didn't ask too many, but I probably did. But thank you again for joining us, Lorenzo. It was really, really great. I can't wait to see what's next coming from your laboratory. And we hope to keep in touch.